Hello.
Good morning, sir.
Morning. Yeah, today I'm taking this class vaccines. Okay, this is my last class on the immunology.
Good afternoon, sir.
Yeah, good afternoon. So, so it's an easy topic, not much to discuss. Already I've sent the presentation. And one important thing is that the same things you have to learn lots of in the second MBBS. In the community medicine, same topic will be taken in community medicine also. These are actually in the community medicine, you will get long questions on these vaccines. So I'll start. You in your already you know lots of things about the vaccines, and importantly, all of us has one or the other day we have exposed to vaccines from your birth. So as you know, this Edward Jenner and Louis Pasteur, they are the pioneers. Do you know what Edward? What is the contribution of Edward Jenner? What he discovered basically?
Smallpox.
Yeah, smallpox vaccine. He was the first person to basically clinically use this smallpox vaccine. So, see, basically, we don't much recognize the vaccines as we recognize the drug. But if you see, if the vaccine is not there, say 50 to 70 years back, there are lots of these diseases like diphtheria, then measles, then pertussis, tetanus. These are the deadly diseases. But nowadays, nobody bothers much about these diseases because the vaccines are there. We don't know much about them. Now everybody is scared about the COVID-19. Once the vaccine comes to the market, then everybody then there will be a common disease like our normal cold, flu. That means common cold. So so before the vaccines are developed, so these diseases are very deadly. But once the vaccines are there in the market, these diseases nobody bothers much. So, if you what is the major challenges in developing vaccines? Like why we are not still now we are able to make this COVID-19 vaccines? The major problem is that say the process itself is a lengthy. You require a long time to develop a particular vaccine, and yeah, and because it is a long time is required, so there are lots of cost is involved, and there is some regulatory clearance also. Regulatory you need ethical clearance, you need DGCA permission. For say United States they require FDA permission. So lots of government permissions are required. And another major drawback in the challenges in this particular vaccine is that many vaccines they are very suitable in the animal models, or whenever they are injected to the cell lines, human cell lines, or animals like rabbit, guinea pig, they can impart immunity. But whenever the same thing it is injected to human individuals, it may not give the equal amount of immunity, and in some cases there's a lots of adverse reactions, that mean unpredictable adverse reactions that may occur in the case of human injection. So these are the major challenges why we are not still able to develop a particular vaccines, say COVID vaccines. And another issue is that if you say if we get some disease, usually we are admitted in hospital, we attend the hospital. But if you see the vaccines are given in the prophylaxis, so it should be available in the in situ. That means it should be available in the community. So making the particular vaccine in the community, maintaining the cold chain, so it requires a lots of cost plus infrastructure. So as a whole, these are the major challenges in particularly developing a particular vaccine. However, with the development of newer immunology techniques, molecular biology techniques, we know that lots of DNA recombinant techniques has come to the market. So, because of the use of this immunology technique, recombinant DNA techniques, they have somehow used the vaccine development. And another issue in this context is the adjuvant. This third point, that is adjuvant. Adjuvants are those chemicals that can impart the effectiveness of a particular vaccine. They increase the effectiveness of a vaccine. Say these are basically chemical substances. Usually they are adult. These are basically added with a particular vaccines. With the vaccine, say effectiveness is say with the vaccine the effectiveness is say 60%. If we add the adjuvant, then it may become say 90% or say 95%. So basically adjuvants basically added the effectiveness of a particular vaccine. And regarding this immunity developed by a particular vaccine, there is basically two types of immunity. The same thing you have probably you have studied in the community medicine, because some classes might have taken by the community medicine also. One is the active immunity and one is the passive immunity. Basically, passive immunity is the principle is that the antibody or the immunity developed from a particular individual can be transferred to the another individual. Best example is maternal to fetal transmission. As you know, breast milk contains lots of antibodies, especially the secretory IgA class of antibody. This IgA class of antibody available in the colostrum of milk imparts the immunity to a particular fetus. Similarly, in your lifetime you must have exposed to tetanus toxoids, okay, tetanus immunoglobulin. So these tetanus immunoglobulin or the rabies immunoglobulin that are basically produced in the horse, guinea pig, rabbit, they can be given to human individuals. So basically the passive immunization is that it don't have memory response. These are basically immunoglobulins that are being transported from one individual to another individuals, okay. But there is no memory response, and this basically immunization is basically a transient. You are giving the antibodies to a particular individual, so it will give a transient immunity, but it don't have this long-term effect since there is no memory response and cell-mediated immune response is not activated. On the other hand, if you see the active immunity, if active immunity is can be imparted by the particular epitope or a particular antigen on a particular individual, like say measles, chickenpox. Usually if we are exposed to measles and chickenpox in the early hour in our childhood, then in future usually we don't again suffer from chickenpox or measles, because the antibody that are being developed because of our natural infection during our early childhood will almost give the lifelong immunity. Or same thing can be done by vaccines. Lots of vaccines are there which by injecting a single dose or maybe sometimes booster dose might be required. They can also activate this particular active immunity. So a difference between active and passive immunity is in passive immunity basically we are transferring the immunoglobulins. So there is no memory response. In case of active immunity, since both cell mediated and then this humoral branch is activated, so there is a memory response, and usually sometimes single dose or sometimes booster dose almost it imparts the immunity, okay. Regarding the passive immunity, like before these vaccines are developed, like before the tetanus toxoids are developed, the development of DNA based rabies vaccines are developed, basically there is lots of use of this tetanus immunoglobulin, then rabies immunoglobulin. However, this use of vaccine, the development of vaccine, somehow it has reduced the use of this particular immunoglobulin. Still we use it, like if a particular individual, we don't know the immune status, whether we don't know whether he has received the tetanus toxoid in his early child according to the national immunization schedule, or maybe if he has not received any tetanus vaccine, tetanus toxoid in last maybe 2 years. So usually we usually give this tetanus immunoglobulin. Or sometimes if there is a say big big injury, like there is a massive injury, then there is a chance that the normal active immunity may not be developed by the time the symptoms will be developed. Similarly, say in case of dog bite, they say grade 2, grade 3 dog bite. So along with this vaccine, usually we give the immunoglobulin so that by the time the active immunity is developed in a particular individual, the immunoglobulin will take care. That's why there are lots of uses of passive immunization. Plus, there are some conditions like anti-snake venom, scorpion bite. Though these venoms, you cannot generate the active immunity in a particular human. So there you have to depend on this particular immunoglobulin. Usually these anti-snake venoms and then anti-scorpion venoms, there are basically these immunoglobulins are developed in the horse serum. Plus sometimes it may be used like in the healthcare workers who are exposed, impending exposure to particular toxins like botulism, Clostridium botulinum. So there you can give this particular immunoglobulin. So though vaccines are there, still there is a lots of uses of immunoglobulins. Then come to what are the drawbacks of the passive immunization? The major drawback of the passive immunization that there is no memory response. So you have given the immunoglobulin, so transiently it will protect you, but long-term protection is not there because it has not developed the cell-mediated immunity. Neither it has activated the B cell response. Memory cell is not there. And another drawback is the allergy. Since you are using a foreign substance, foreign serum, that is basically this, as you know, these immunoglobulins are developed either in the horse or maybe in rabbit or maybe in guinea pig. So it is a foreign to our human individual. So it may cause the type 1 anaphylaxis reaction. And as you know, the type 3, that serum sickness, this is basically type 3 reactions. This is immune complexes basically mediated by the neutrophils. So this type 3 reaction, the serum sickness, basically it is done by these vaccines, especially those immunoglobulins. So main problem is that they don't have the memory response, and second thing is the hypersensitive reaction, especially the type 1 and type 3 hypersensitive reactions. However, this development of newer molecular biology techniques, we can, the whatever antibodies that are being developed in a particular horse or maybe in rabbit, they are humanized. Humanized means the portion that will be, especially the FC portion of the immunoglobulin, that means the constant portion of the immunoglobulin has been replaced by, spliced by human FC fragment using the molecular biology technique. So ultimately the variable region will be the same. Variable region will be basically developed from the either from horse or maybe rabbit or maybe guinea pig, but the FC portion has been replaced by the human one. So that is called humanized immunoglobulins, monoclonal antibodies. Then come to the active immunity. Already I have told that active immunity, basically there is two types of active immunity, either by natural infection or by injecting. Now, if you see the active immunization, that is the best immunization techniques, because there is a memory response. Both B cell and T cell is activated. So both antibody, that means both humoral and cell mediated immunity is developed. So ultimately it gives a more effective immunization against a particular pathogen. Now, come to the designing of a particular vaccine. Now, if we design a particular vaccine, then what are the important things that we have to look in a particular development of a particular vaccine? One of the important challenges, or you can say in the designing a particular vaccine, is that our aim is to activate both T cell and B cell, so that memory response is good, both the humoral and cell mediated immune response is activated, and it gives the more immune response. So while designing a particular vaccine, we have to give one important thing, is that our aim should be to activate both the T cell and B cell, so that immunity is good, memory response is there. But still there are some limitations. Like say in the book it is written that normal cold, common cold, that is by Haemophilus influenzae, their incubation period is only 1 to 2 days. That means within 48 hours the symptoms are developed. So if you depend on the active immunization, your memory cell may not get time to develop your antibodies, or may not develop may not have the time to develop a particular T cell, activated T cell. So in this case, what you have to do, you have to give repeated injections of a particular vaccine. Like influenza vaccines, they are in Western countries, they are given in yearly basis. Per year, this vaccine is given, this influenza vaccine, so that this vaccine can generate the antibodies, and this antibody, somehow we are maintaining a steady concentration of the antibodies. Because if we only depend on the memory cells, then memory cells may not have time to develop the antibody by the time symptoms are developed. So the major limitation is the incubation period. So those in the organisms which have a very short incubation period, like the Haemophilus influenzae, only 1 or 2 days, there you have to go for repeated injections, so that you are maintaining a particular concentration of the antibodies. However, if the incubation period is long, say poliovirus, where the symptoms never develops before 3 days, their memory cell can take care. So that's why in case of this polio vaccine, say you don't require repeated injections or repeated infusions in per year basis. So regarding the types of vaccine, this is I have made it. It is not. It is there. The types are there in the book. So this is the major classes of a particular vaccines. This is either it is maybe live attenuated or inactivated, and there may be some purified macromolecules. So we'll discuss each and every types of these vaccines. Live attenuated vaccines are basically, these are live organisms. Inactive or kill, name itself signifies that these are the kill vaccines. Then there are purified macromolecules. That means in purified macromolecules, whole virus or whole pathogen we are not using, only a part we are using, like polysaccharide capsule, or maybe toxin in case of toxoid. Plus the last one, the recombinant DNA antigens, there you can again subclass into 3 headings: purified proteins, recombinant DNA vector vaccines, and finally DNA vaccines. So we'll discuss one by one. Okay, if we consider the major types of vaccine, you can consider the live, inactive or killed, and purified proteins. In the purified proteins, again 3 classes you can consider: polysaccharide capsule or toxoid, and recombinant DNA antigens. And recombinant DNA antigen again sub-classified to another 3 subclasses: purified proteins, recombinant vector vaccines, and DNA vaccines. So we'll discuss one by one. Live attenuated vaccines. Sometimes in the book you will find attenuated vaccines. There may not be this word live. So live attenuated vaccines are basically those vaccines where the organism is living, or say virus or pathogen in the vaccine is living, but the virus or the bacteria is grown in some adverse conditions. Because they are grown in adverse conditions, they don't have the pathogenicity. That means they cannot produce the disease, but they have the antigenicity. That means they can induce the immune response. So if you select a particular organism and you grow in an adverse medium, that is it's not natural medium, so there will be some mutant forms are developed. These mutant forms will ultimately grow in your culture medium, because you are using a adverse artificial medium. So that mutant form don't have the same effect in the natural host. Like Robert Koch. Robert Koch discovered these BCG vaccines. As you know, the tuberculosis basically caused by Mycobacterium tuberculosis. So in this BCG vaccine, Bacillus calmette-guerin, so in this BCG vaccine, what Robert Koch did, he selected one particular Mycobacterium bovis strain. Okay, this Mycobacterium bovis strain, he cultured in an increasing concentration of bile and cultured for 13 years. That means 13 years, this Mycobacterium bovis is cultured in adverse conditions of bile, increasing concentrations of bile. Because they are repeatedly cultured in these adverse conditions in the bile for 13 years, they have lost the pathogenicity. That means they cannot cause the disease. That means they cannot cause the tuberculosis, but they retain the antigenicity. That means their immune response is okay. They can generate the immune response. Similarly, Sabin polio vaccine, that is normally we call it a oral polio vaccine. You know that OPV or oral polio vaccine is nothing. This is the Sabin polio vaccine. It is grown in the monkey kidney epithelial cells. Similarly, in the last one you can see that the rubella virus, that is grown in duck embryo cells. So and later on human cell lines. So these are the basically pathogens are basically grown in the adverse conditions. That's why they have retained the antigenicity, but they have lost the pathogenicity. That means they cannot cause the disease. So what is the advantage of the attenuated vaccines? If you see the vaccines, the classes, different classes of vaccine, there are some distinct advantages of live attenuated vaccines. Live attenuated vaccines, since the virus or organism is living, that means they can replicate in the host. Okay, they can replicate in the host. Say you are giving BCG vaccine to a particular individual, they can this Mycobacterium bovis can replicate. Since they are replicating, so their epitopes are exposed to our immune systems. So our immunity is developed against the different epitopes of a particular organisms. Since they are attenuated, so they cannot cause the disease. So these epitopes will produce that means these antigens will produce the immune response, and both the T cell and B cell is developed or activated and leading to the generation of memory response. That's why, since the memory response is there, the live attenuated vaccines usually we don't require booster doses. Usually single doses, most of the cases single doses is almost sufficient. Like BCG vaccine, if you know the national immunization schedule, BCG vaccine is only given at the birth. Okay, because they are a live attenuated vaccine, so they can replicate in the particular host, and because they are replicating, so all epitopes are exposed to our immune cells, and both T cell and B cells are activated. Memory response is good, so we don't require much booster doses. However, this oral polio vaccine, Sabin polio vaccine, is a bit exceptional. You know that this is given in 3-4 doses. Usually the problem with the Sabin polio vaccine is that there are basically 3 strains of the virus. Say we are considering the 3 strains like A, B and C. Now whenever you are giving the oral polio vaccine or Sabin polio vaccines, one of the virus will replicate in the gut. Say out of A, B, and C, A has replicated. If A has replicated, so A has grown. So our immune system will develop antibodies against A. Okay. Next time, whenever again injecting or not injecting means oral polio vaccine, whenever you are giving the oral polio vaccine in the gut, since antibodies are developed against A, next time this oral the virus A will not able to grow because already we have developed the antibodies against A. So this time say B will replicate. Now since B is replicating, so our antibodies will develop against B. Okay, so now we have antibodies against A and B. So whenever you are giving the this oral polio vaccine third time, then since antibodies are both there against A and B, only C will able to replicate. So C will grow. Because C will replicating, C is growing. So antibodies again it will be developed against the C. In this way, in 3 doses, the immunity is developed against all the 3 viruses. Why it occurs? Because this particular vaccine contains 3 strains of virus. Another important thing I forgot to mention in this particular live attenuated vaccine is the herd immunity. That means immunity to a particular community. Say you are giving oral polio vaccine to a particular baby or particular children. Along with the feces, the live attenuated virus will be mixed with the water. So if it is mixed with the water, so that virus will be distributed in the community. So that virus may be again the virus can be again ingested by a particular non-immunized children. So that virus, since it is a live attenuated virus, so non-immunized children indirectly he is getting this particular attenuated strain. So ultimately, though we are not giving the vaccine to a particular non-immunized children, but because this is a live attenuated strain, so the particular children has got it from for the contamination from the community. So this is known as herd immunity. Another issue is that in the live attenuated strain, say polio vaccine, this is a attenuated strain. This particular virus is replicating in the gut or vaccinated children, then it is excreted in the feces. So this virus is mixed up in the natural environment. So this virus will now compete with the normal pathogenic virus. So gradually it will gradually shift the normal pathogenic virus, and because of the competition, there will be competition between this attenuated virus and pathogenic virus, and gradually the pathogenic virus will be thrown away from the community or the environment. So lots of advantages of attenuated vaccines are there, but still there is some disadvantage because these are the live vaccines. That means the organism is living. So there is every chance that though it is a remote possibility, but there is a chance that the virus can again mutate to the original pathogenic virus. Like in case book, it is written that every 1 in 2.4 million doses of this particular oral polio vaccine or 7 polio vaccine, there is a 1 in 2.4 million conversion. That means if you give 2.4 million doses of this particular vaccine, in one case, the virus can be again mutated to a pathogenic one. And sometimes, like some vaccines, especially the measles, if you are giving this live attenuated vaccine to a particular individual, sometimes the symptoms are developed and symptoms are similar to that of the natural virus, like rubella. Measles vaccine. That's measles. If you give them measles vaccine, sometimes it may happen this measles vaccine itself causes your hepatic encephalopathy. So either the disadvantage is that sometimes the attenuated virus can be mutated to the natural virus, and sometimes the symptoms because of the infusion or injection of the particular live attenuated vaccine, it can produce the symptoms that are natural to a particular pathogen, like measles vaccine. Measles also can produce the encephalitis. The same vaccine can also produce the post-vaccine encephalitis. However, lots of genetic engineering techniques has come, like herpes virus. That is the pig herpes virus. This pig herpes virus, here thymidine kinase gene is removed. So if you remove the thymidine kinase virus, and inject this particular attenuated strain to human, it cannot grow in the neurons because for grow in the neurons they require the particular enzyme thymidine kinase. Similarly, in case of rotavirus, here live attenuated virus is basically taken from animal virus. In the animal virus, by genetic engineering, you are putting some antigens from a virus that causes the human one. That means we are selecting some animal viruses, animal rotavirus, and we are basically by using the genetic engineering, we are putting some antigens from the human one. So using the genetic engineering techniques, however, the live use of these live attenuated vaccines has these are mostly lots of live attenuated vaccines are being developed by using these particular genetic engineering techniques. Then come to the inactive or kill vaccine. As the name signifies, kill vaccine, these are not live vaccines. That means these are the organism is killed. It may be bacteria or maybe virus. Now killing is basically done by either by heat or any chemical substance like formaldehyde. Now as the name signifies, these are the inactivated or kill vaccines. So either you can use heat or chemical substances. But if you see the heat, if you heat the particular protein, basically this, you know that these tertiary, secondary, quaternary structures are broken. And if the tertiary, quaternary structures are broken, there is an extensive breakdown of this tertiary and quaternary structure. So normal epitopes cannot develop the immune response in a particular individual. That's why usually nowadays this heat inactivation is not used. Mostly it is used the chemical inactivation like formaldehyde. Like there is, in case of polio vaccine, there is two types of polio vaccine. One is the oral polio vaccine, that is already I have discussed in the 7 polio vaccine, and another is a shock polio vaccines. In the Western countries, basically this shock polio vaccine is being used. There basically the polio virus is inactivated by using the formaldehyde. So what are the drawbacks or advantages of this particular inactivated vaccine? Inactivated vaccines are, you don't require repeated cultures like BCG vaccine. It requires 13 years to develop a particular attenuated strain. So inactivated kill vaccines you can generate in a very early short time. But there is problem is that they are not much effective against the cellular response. The cellular immunity is not much developed. Mostly it is the basically B cell, the humoral branches mostly it is developed. And since the cellular response is inactive, so memory response is very less in case of this kill vaccine. So you require a booster doses. You have to give booster doses infrequently. You have to give in doses of this particular kill vaccines. But however, advantage is that you can, in a short duration, you can develop these particular vaccines. And sometimes if you are by using the chemical substance, if you are not able to kill or inactive the particular virus, then the virus can cause the natural disease. Like in the book, it is written that whenever the shock polio vaccine was developed, in some vaccines the virus failed to kill, the formaldehyde failed to kill the virus, and leading to this particular sign and symptoms of the polio. So these are the advantages and disadvantages of inactivated vaccines. Then last one, purified macromolecules. In the live attenuated vaccines or kill vaccine, we are using the virus or pathogen as a whole. Here, we are not using the whole pathogen. We are using a part of the pathogen. So basically, it can be exotoxins, inactivated exotoxin, we normally call the toxoid, capsular polysaccharide, and finally the recombinant microbial antigens. So let's discuss one by one. Bacterial polysaccharide capsules. You know that lots of bacteria, basically they produce the pathogenesis from the lipopolysaccharide core. In the capsule, they have some lipopolysaccharide core which will inhibit the immune response. So our aim here is to develop antibodies with this particular lipopolysaccharide antigens. Like if you develop antibodies against the lipopolysaccharide antigens, this antibody will go and coat or opsonize a particular bacteria, and these opsonized or coat bacteria can be removed from the circulation by reticuloendothelial systems, or you can say macrophages. However, there is some limitations of this particular bacterial polysaccharide capsule is that they are not much able to develop this cellular response. Their memory response is not there. They are not much effective in developing the helper T cells, and mostly IgM class of antibodies are developed, and there is little class switching. So IgG class of antibodies are not produced. Mostly the IgM class of antibodies are produced in case of bacterial polysaccharide core. Then another is the inactivated exotoxin or toxoids. As you know, there's titinous, basically we call TT, that means titinous toxoids. Basically toxoids are what? Toxoids are basically toxins produced by a particular bacteria, like Clostridium tetani. Clostridium tetani produces the toxin, and this toxin is inactivated and being used as a vaccine, and that is known as titinous toxoid. So here, basically the toxins from a particular bacteria or pathogen is taken, and it is inactivated by a particular chemical process, and that inactivated toxin can be injected as a vaccine. So so major limitation of this particular inactivated toxin is that because we need the toxin, so there is a requirement of a huge amount of toxins which has to be inactivated, and there is a limitation in this particular toxin is that we don't have much of these toxins, because the toxin is produced by the particular bacteria whenever they infect a particular natural host. Okay, so here basically whole bacteria we are not using, we are using a particular toxin of the bacteria. That's why that toxin only will get whenever the bacteria will infect a particular individual. And like other inactivated inactivated capsular antigen, they are also less in generating the memory response. Then recombinant DNA vaccines. So now because the DNA recombinant technology has come into the play, so we know that which one is the DNA sequence of a particular pathogenic antigen. Say if we know the antigen, then we can get the sequence or the gene for the particular antigen, and gene can be incorporated to a particular bacteria or yeast or insect, or maybe in case of mammalian expression factors. And whenever this particular recombinant DNA is expressed to a particular expression factor, ultimately you can get this particular protein, and you can use as a vaccine. Like in the book, it is the best example of this hepatitis B vaccine. Hepatitis B vaccine is based on the recombinant DNA technology. Here, what do we do? The surface antigen gene is used to develop this particular vaccine. What we do, the gene for the surface antigen is expressed in a particular yeast cell. Now, like yeast can be fermented. Yeast can be fermented in a large fermenter. Whenever yeast will go, it will along with it will express the surface antigen also. There will be lots of surface antigen protein, and this surface antigen protein can be again purified by different purification technique like HPLC, and that can be used as a generation of the vaccines. Before developing this recombinant DNA technology, previously there is a use of these synthetic peptides. Since you know the pathogenic antigen, so you know the DNA sequence of this particular pathogenic antigen, so you can synthesize a peptide based on this particular sequence, and this peptide you can use as a vaccine. But the problem is that, as you know, that proper immune response by a particular antigen depends on the proper conformation, like secondary and tertiary structure. So even you synthesize a particular synthetic peptide, it may not have a natural conformation. It may not have a normal secondary and tertiary structure that being available for a natural one. So it may not impart the normal immune and humoral and immune cellular branches of the immunity. So that's why they are less effective in developing a particular immune response. So nowadays, that's why nobody uses the synthetic peptide to develop the vaccines. Then recombinant vector vaccines. In the recombinant vector vaccine, what we use some attenuated strain. We use some attenuated strain to carry the antigen of a particular virulent strain. Like In the book, you will find that attenuated strain of Salmonella typhimurium. Here, it has been engineered with genes from the Vibrio cholerae. So the Salmonella typhimurium is the attenuated strain. That means it cannot produce the pathogenicity in case of human individuals. And with this attenuated strain, if you engineer with genes from the Vibrio cholerae, that means it will carrying the genes of the Vibrio cholerae. So in the gut, the genes of the Vibrio cholerae will express. So it will express the protein. So ultimately, the immunity is also developed against the cholera. Similarly, the another is the vaccinia virus. Vaccinia virus is basically it is the attenuated strain of the smallpox. Smallpox has already been eradicated from the world. So what will do with the attenuated strain? So vaccinia virus is used as a vector. So basically, in this particular recombinant vector vaccines, we are using some live attenuated strains to carry the antigens from a other pathogenic organism. Like how to use a particular recombinant vector based vaccine. Here we use some plasmids. Say we have in this plasmid we have incorporated the pathogenic gene of a particular pathogen. Okay, and then this recombinant plasmid we can combine with a particular vector vaccine like vaccinia virus. So in this vaccinia virus, if we grow in a culture medium, so it will have this recombinant plasmid also. So recombinant plasmid, since we are carrying some amount of this pathogenic antigen, so this pathogenic antigen will also express, and ultimately the as a whole immune response will be developed. And finally, the DNA vaccines. DNA vaccines, these are basically, here we are not using any vector. Here directly we are incorporating the DNA. If we know the pathogenic form of that particular, if we know the DNA sequence of a particular antigen, so we can synthesize a particular sequence of the DNA, and that DNA can be directly injected. So if you directly inject that DNA, the muscle cells will take some will be taken by the dendritic cells. So whenever muscle cell or dendritic cell will take this DNA, it will be incorporated with the normal chromosomes. Either it can be incorporated in the chromosome, or it may be it can be like in the episomal form also. So if it is incorporated to the human chromosome, so ultimately that sequence, that antigen from the pathogen will express the protein. So the protein, since the expression of protein will occur, so gradually there is a generation of the immune response. So both T cell and B cell will be activated. So there will be a good amount of memory response. Another advantage of the DNA vaccines is that we don't require the booster doses. Since the DNA has been already incorporated to the chromosomal form, so it will lie in the chromosome. So it will continuously express the particular protein. So here what we use some gold particles. With those particles we coat with particular plasmid DNA, and this plasmid DNA will consist some of the antigens from the pathogen, and it will be incorporated in the muscle cell using a zinc gun. However, this DNA vaccine till now it has not come to the market. Basically, it has been shown successfully in the animal models only. In the animal models, these DNA vaccines are very much effective, but till now in the for human use, it has not come to the market. So lots of advantage of these DNA vaccines are there. Like since these are the DNA vaccines, so you don't require the booster doses. Since DNAs are very much stable at the temperature, so you don't require the cold chain to be maintained. So refrigeration is not required. So you don't require a booster doses. Single dose is sufficient. If you incorporate that particular DNA in the chromosome, you don't require booster doses. It will lifelong. It will stay. But there is some also disadvantage that you cannot revert back, since the sequence, the gene, you have incorporated to the chromosome, so you cannot again revert back like the other ones. You cannot revert back the immunity. Another issue is that many of the pathogens, they are carrying the polysaccharide core, lipopolysaccharide core. Now lipopolysaccharide and polysaccharide antigens, because these are carbohydrate based, so how will you produce the DNA type of vaccine? You cannot produce DNA vaccines for polysaccharide and lipopolysaccharide antigens. So these are some drawbacks of these particular DNA vaccines. So I will come to these basic classifications. Yeah. live attenuated vaccines like oral polio vaccines. Advantage is that these are live vaccines. Virus or pathogen can grow. Immunity is good. Both T cell and B cell is activated. Memory cell is there. But the drawback is that since it's a live virus, so it can be again mutated to the pathogenic one. And sometimes these particular live attenuated vaccines can cause the natural disease if it is again mutated. Inactive or killed vaccines, since you have killed it, so your immunity will be a bit less. Basically, they will mainly impart the humoral branch. Cellular immunity is a bit less. That's why memory response is less. You require lots of booster doses. Repeated injection may be required. Whereas live attenuated one, since they are live one, so they have impart the immunity. They can replicate in the natural host. So you don't require much repeated infusions. Usually single doses is sufficient. Purified macromolecules, biggest advantage is that you are not using the pathogen as a whole. So there is no chance of the pathogen can cause the disease because you are using a part of the pathogen. Say in polysaccharide capsule, if you are using polysaccharide capsule, advantage is that there is no problem of the causing the disease. But the drawback is since you are only using the polysaccharide capsule, so you are activating the immune response, the basically the humoral branch. Only IgM class of antibodies are developed. IgG you are not able to develop. Class switching is not there. Cellular response, you may not have this equal amount of cellular response. Toxoid like tetanus toxoid. So in this, or like diphtheria toxoids. So in this toxoid, basically we are using the inactivated toxins. We are not using the pathogen. We are not using the Clostridium tetani. We are not using the pathogen. We are using these particular toxins to develop the vaccine. So here, the major disadvantage is that here also immunity is not much. Memory response is a bit less, and you don't get much of the purified toxins, since the toxins are produced whenever they will infect a natural host. Last one is the recombinant DNA antigen. All these are based on the recombinant DNA based technology. Purified proteins like your hepatitis B vaccine surface antigen. Here the advantage also is that you are not using much of that whole organism. You are not using the whole organism. But here there is also some problem, like here purified proteins, whenever you are using purified protein like surface antigen, that virus not only carrying the surface antigen, it is also carrying the other antigens also. So you have to make the vaccines for the other antigens also. Then recombinant vector based vaccines, these are basically here live attenuated strains are used, and by genetic engineering you are incorporating some antigens from a particular pathogenic organism. And finally, it is the DNA vaccines. Theoretically, DNA vaccine should be the highest effectiveness, because if you allow the DNA vaccine to come in the market, and if the DNA vaccine comes to the market, the DNA of the particular antigen of a particular pathogen, if it is incorporated to a chromosome, then whole life the particular antigen can be expressed and it can impart the immunity. So you don't require booster doses. However, these are only success till now. It are successful in the animal models. In the human models, it has not come in the market. And another drawback is that these DNA vaccines you cannot use for lipopolysaccharide core or say polysaccharide core, because for DNA vaccine to be made, the antigen should be a protein in nature. It cannot be a carbohydrate in nature. So these are the advantages and disadvantages of our different types of vaccines. So this chapter is a bit easy, not much to discuss. Almost, I think mostly you will get short notes in the exam from this one. And same topics again, it will be discussed in community medicine also. So any question from these vaccines? So students, any question? If there is no question, I will stop it here. Okay, then I will stop it here. And as such, officially, I will not take any more classes. If you require, you let me know if any classes or any topic is required to be again revised. Because as per my syllabus, all topics I have covered. And one topic, previously I used to teach complement, but in this MCA syllabus, this complement is not there. So I'll stop it here. If there is any confusion, you let me know. And any topic has to be revised, you let me know. I will take some day. Okay. Okay. Thank you.
Thank you, sir.
Thank you, sir.
Right now. I